Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, Impact of PTEN Protein Expression on Benefit from Adjuvant Trastuzumab in Early Stage HER2-Positive Breast Cancer in NCCTG Adjuvant Trial N9831 by Edith A. Perez et al. My name is Hope Rugo, and I am a professor of medicine at the University of California, San Francisco Comprehensive Cancer Center in San Francisco, California, USA. My oncologic specialty is breast cancer. As options for the treatment of HER2-positive breast cancer continue to expand, the search for markers that predict benefit from specific targeted agents has become a critical research goal. Ideally, these markers would predict benefit from specific agents rather than a class of therapy. For example, in the case of HER2-positive breast cancer, the first step would be to identify a marker that predicts lack of benefit from trastuzumab, or those that predict benefit from the use of additional HER2-targeted therapy. A number of studies have identified markers that are prognostic, correlating with worse disease outcome, but identifying predictive markers for individual therapeutics has proved to be more elusive. Phosphatase and tensin homolog, PTEN, is a phosphatase protein encoded by the PTEN gene that acts as a tumor suppressor gene, playing a key role in cell cycle regulation. PTEN is one of the most commonly altered tumor suppressors in cancer, with mutations and deletions that cause reduced activity of its enzyme, with resulting loss of regulation of cell proliferation. Preclinical data suggests that loss of PTEN results in upregulation of signaling through the phosphatidylinositol-3 kinase, or PI3 kinase AKT pathway, and therefore relative resistance to trastuzumab. Small clinical series have provided further evidence that loss of PTEN is associated with low response rates to trastuzumab. In addition, due to its different mechanism of action, it was initially postulated that PTEN loss might predict relatively better efficacy of the dual kinase inhibitor lapatinib compared to trastuzumab. Biomarker studies in inflammatory disease and in the neoadjuvant setting failed to demonstrate any conclusive association between alterations in PTEN or activation of PI3 kinase and activity of a specific agent or class of agent. In reviewing these data, it is important to note that small or single-arm series such as these are hypothesis-generating, but are more likely to describe potential prognostic rather than predictive markers. In this study, investigators evaluated expression of PTEN in patients with early-stage breast cancer randomized to chemotherapy with or without trastuzumab in the U.S. cooperative group study N9831. Tumor samples were available for a subset of patients. 37% had tissue microarrays from tumor blocks, and 15% had only tissue sections due to insufficient tumor material, representing just over 50% of the entire study population. PTEN protein expression was evaluated using antibody staining by immunohistochemistry, or IHC, and scored based on intensity of cytoplasmic staining. At least some degree of PTEN expression was seen in 3/4 of patients. and although there was a modest association with expression and node positivity, as well as absence of hormone receptors, there was no impact on overall disease-free survival. In addition, there was no difference in disease-free survival comparing patients treated with trastuzumab or not, based on presence or absence of expression of PTEN, or based on intensity of staining, even when staining was grouped into several different categories. Based on this data, expression of PTEN is neither prognostic nor predictive in patients with HER2-positive breast cancer. Interpretation of this data requires consideration of the testing and scoring methods employed. There is no standard test for PTEN expression, and loss of expression may not always correlate with absent enzyme activity. We are hampered necessarily in exploratory biomarker studies with a lack of definition of what actually defines abnormal. The investigators tried to make up for this lack of information by evaluating various combinations of intensity of IHC staining without finding an association with response to trastuzumab or with outcome in the entire trial population. The extent of heterogeneity of both predictive and prognostic biomarkers in tumors is beginning to be understood and is difficult to account for in the setting of a clinical trial. Interestingly, in this analysis, significant heterogeneity was found in PTEN expression, with agreement between scoring of TMAs seen in only about half of duplicate samples. This discordance impacts the ability to assess the impact of expression as both a prognostic or a predictive marker. At the San Antonio Breast Cancer Meeting in December of 2012, Jose Baselga reported biomarker data from the Cleopatra trial, which randomized patients with chemotherapy-naive metastatic HER2-positive breast cancer to receive docetaxel with trastuzumab and either placebo or pertuzumab, and demonstrated a 38% relative improvement in progression-free survival, as well as a significant improvement in overall survival with the addition of pertuzumab. Multiple markers important to signaling through the HER2 pathway were evaluated in various subsets of patients and in both tissue and serum. None showed a correlation with differential benefit from pertuzumab. Indeed, although specific markers correlated with outcome, in particular mutations in phosphatidylinositol 3 kinase, or PI3 kinase, this was independent of treatment arm, and all subjects appeared to benefit from the addition of pertuzumab. These data are consistent with previously presented biomarker analyses from the neoadjuvant NEOSPHERE study, as well as other smaller trials. These data, taken together, lead to the conclusion that, to date, HER2 overexpression is the only marker available for predicting benefit from HER2-targeted therapy regardless of the agent. For a disease subgroup with multiple active agents, for now we are the victims of our own success. Targeted therapy for HER2-positive breast cancer works well indiscriminately, is associated with only modest toxicity, and more appears to be better. In order to understand what tumors or patients require more, or perhaps less, than chemotherapy combined with trastuzumab for optimal outcome, we will need to identify a panel of markers or gene signatures rather than focusing on specific proteins. Ongoing studies should lead us closer to this goal. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.